Dear audience, it gives me immense pleasure to introduce respected, highly intellectual, Padma Bhushan awarded Professor Nirmal Kumar Ganguly as the chief guest of today's event. Sir is the former Director General ICMR and was a distinguished biotechnology research professor at Department of Biotechnology, Government of India. Sir has published more than 775 research papers and has supervised 130 PhD theses. In 2015, he was selected for award of Helmholtz International Fellow by Helmholtz Center for Infection Research, Germany, for his significant contribution to the field of infectious diseases. Besides this, he has received 7 international and 111 national awards, including the prestigious Padma Bhushan Award in 2008 in the field of medicine. A big round of applause for him, audience. So now is the time for the most awaited address by our chief guest, Professor N.K. Ganguly. A huge round of applause for him, everyone.
University, Professor Rajveer Singh, Honorable Vice Chancellor of Punjab, Haryana Central University, Professor Kuhar, Professor N.K. Arora, Professor Parveen Rishi, and Professor Singh, and all other dignitaries over here, very distinguished scientists sitting in the audience. It has been a great pleasure to come here to be amongst you. I remember I attended the first AMI meeting in Hisar around 45 years back. I don't remember the year. That was also held in Haryana only. It was held in the Hisar University. So today, I will be talking about a small piece of research which was conducted recently. But before that, I want to say that I'm so happy that Parveen has become the president of the society. She was my PhD student in PGI Chandigarh, and it gives me great pleasure to see her here taking the mantle of the society. I will be presenting a very small piece of my recent work about the microbiomes, that how the beneficial microbiomes or the bad microbiomes affect our health. We have more microbial cells than our cells in our body, populating our skin, mucosal surfaces, even the eyes. And we have recently learned that they actually control every aspect of our bodily function, whether boosting the immune system, bringing down the inflammation, creating controlling the metabolic health, as well as providing a lot of proteins and peptides and others which have either beneficial functions or deleterious functions. So if we really look at that, many of the bad microbes actually are responsible for our bad health. And one of the things which now has been identified as a big disease, that is dysbiosis, which is caused by these bad microbes. So I will give you a little bit of vignette about how the microbes help in the cancer. So if you move the slide. So these are my collaborators. The main work has been done by Kriti Jain, my PhD student, and the other collaborators are Amitav Awasthi, Shyam Agarwal, Rashmi Rana, Deepak Bhavtosh, and Surajit Ganguly, and Parul has analyzed it. And Jassi has helped me in doing the metabolomes. He is in the ILBS. Next, please. So, what happens in our body when you have cancer? It evades the immune system. So, to complete an immunologic reaction, there is a primary reaction which happens with the CD3, and there is a secondary reaction which completes the immune reaction, which is with the co-stimulatory molecules. But when the cancer happens, these co-stimulatory molecules are replaced by proteins of similar structure, the PDL and PDL1, which actually create a checkpoint so that the immune system doesn't recognize the cancer and cancer thrives and grows. So one of the very revolutionary therapy which came up was the checkpoint inhibitors. When you give the checkpoint inhibitors, it allows the immune cells to recognize the cancer, develop and expand the clones, and then attack the cancer. So this is the hypothesis. So what happens that when the mechanism of blockade happens, the immune cells become exhausted and tumor-induced immune suppression happens, and tumor progresses. Next, please. And so the checkpoint inhibitors which have happened, there are about 6 or 7 different checkpoint. Next, please. There are 6 or 7 different checkpoint inhibitors which are used. We have used the cheapest one, which is the nivolumab over here. But the drawback is this, that these are frightfully expensive. One shot about 78,000 or something, and only 30 to 35% respond to the therapy. Rest do not respond to the therapy. So if we could know that what are the biomarkers where we could recognize who will respond and who will not respond, then we could actually spare the persons who will not respond with this expensive therapy. They almost sell their home and hearth to get this. So present biomarkers are about 3. The PDL expression, if it is more than 30%, it is helpful. The tumor mucosal burden, mutation burden, the more tumor mutation happens, there are more neoantigens, which the immune cells could recognize, and microsatellite instability. These are at the moment, but these have a limitations. When we checked in our study all of these, we found that they were not able to differentiate responders from non-responders effectively. So our goal was to identify these biomarkers. Next, please. So we actually looked at the entire system which I just talked about in this particular study. So the responders and non-responders, the non-responders had a progressive disease, and responders had either complete or partial or stable disease. Either they responded completely or partially or had a stable disease. Next, please. So this is how our samples came. We have, from the PBMC, we did about 60 immunologic panels. With the plasma, we did the proteomes and microbiomes using the Orbitrap. From the serum, we looked at about 40 cytokines through the Luminex system. And with the tissue, we looked at the whole genome sequence. And the fecal samples, we looked at the microbiome. So microbiome is the part which I will today present. And I will also present that how the metabolic pathways and the proteins are altered by that. The upper part of the studies I will not present because the time will not permit that. Next, please. So they were given 3 injections about 2 weeks apart, and they were followed up to 5 years. So we have 5 years follow-up samples. So in this, I will be happy to say that normally lung cancer subjects, they die around 6 months to 9 months or an year. Majority of them, except few, where targeted treatment is available. In our series, 50% of these they have survived for the 5 years follow-up which we had. Next, please. So I will just go direct to these are the different things which we did for metabolomes. Next, please. the methodology. If we look at, there are two analyses which have been done here, principal component analysis and partial component, partial least square determinants. And here we look at the two parts for the healthy, for the cancer and controlled subjects, and we find that they separate very, at the baseline they separate very nicely, and you could differentiate them. Next, please. This is a heat map. The green part are the responders, and the other red part are the non-responders. We had put up hierarchical clustering analysis of top 20 metabolomes over here. So 50 of these metabolomes were identified. Next, please. And this is a volcanoplat. You could see the green ones which have come down and the red ones or the brown ones which have gone up. So there were really very, very nice differentiation between all these. Next, please. So this is between the same analysis between the lung cancer subjects and the controls. And again, the same differentiation has happened. Next, please. And this is a heat map, so showing the hierarchical differences between the two. The green ones responders, red ones non-responders. Next, please. So now with the reduction analysis, this is the KEGG pathway where you actually normalize the plots of the metabolomes which are significant, and we see nice curve which is moving up, and we could identify the strongest one which are important in the metabolic functions in the cancer. Next, please. So this is the crux of the thing, because I will not describe all the 50 ones. With the reductions approach, we got sphingolipid metabolic pathways, arachidonic metabolic pathways which control the inflammation, and glutamate metabolic pathways. The sphingolipid induces tumor cell proliferation, resistance to chemotherapy, radiotherapy, immunotherapy, and induces metastasis. So it's a bad thing. And there are a lot of different pathways which are controlled by this, including the AKT. The arachidonic pathway provides some inflammation, which sometimes is beneficial, sometimes is not. But glutamate metabolism actually is very beneficial. Glutamate metabolism reconditions the tumor microenvironment and can enhance the anti-cancer immune response through both direct and indirect mechanisms. Next, please. So this is again the KEGG analysis. Next, please. So here we can see some of the pathways which are coming out of that. In this, we can see that urea pathway is one of the best one which is found in the responders. Very nice. But hydroxybutanol, gentisic acid, etc., sphingolysine, etc., are found more in the non-responders. The red ones are non-responders. Next, please. So this is again the glutathione metabolism which was published in JCV. Next, please. So the urea cyclase dysregulation generates clinically relevant genomic and biochemical signatures. I will show you the data next in the data. Next, please. So if we look at the ROC curve, we see that both the area under the curve as well as the sensitivity specificity is about 100% to 98%. Next. If we take the overall things, specificity comes down a little bit, but it still is very, very relevant. Next, please. And if we look at the various Kaplan-Meier analysis, we find the survival plots. Next, please. All the survival plots with the different things show the same thing. There is a very good 50% survival. Next, please. So all these are different survival plots. Next. So now I come to the microbiome, which is the subject of today. So we did alpha diversity analysis. The block spots, this block spot shows the evenness, both Shannon index, Shao index, with a Wilcoxon test, has been used over here. There was no significance, but this shows that the data is robust. Next, please. So this is the heat map, and the various organisms are shown here. The green one again are responders, red one are non-responders, but the next one will be much more clearer. Next, please. These were all done through the complete sequencing of the things. So in the responders, we find the bifidobacterium is important, the Doria is important, one of the unnamed taxonomical bacteria which we have given a number was important, and Polyphomonas were important. However, in the non-responders, the Enterobacters as well as the Klebsiella were in abundance. Next please. So in this heat map, again we see the same thing. Next please. So these were the various functional analysis which were done through the metabolic pathway, but I will show you the final structure. We'll skip few of the slides. Next please. Next. So here the red lines show we have done the complete metabolic pathway with the organisms, which you can see here, and the correlation network shows the more thicker it is, the better correlation. But the blue ones are in the non-responders, and the red one are found in the responders. So these microbiomes are actually controlling the complete metabolic pathway in our patients. Next, please. So you see the correlation, beautiful correlation. Next. So we did some targeted proteomes. I will not bother you with it. This is a very new technology, and we could see that there were about 5 different targeted proteins which were different. So in this analysis, we found IL-13, Granzyme A, VGF2, TWIG, GZMH, and CD27 significantly change over time in the non-responders. And then responders, the CRTM, CCL23, and Granzyme 2 actually changed. Next. And if we look at the temporal changes in the responders, this is very important when you follow up the patients, whether they're responding, and there was a very good correlation. Next. So the final slide, next please. The final slide shows that how the metabolic signatures are different in non-responders and responders, how the gut microbiome are different in the responders and non-responders, and how the proteomic signatures are also different in responders and non-responders, and many of them are being controlled by the microbiome which are inhabiting in our gut. Now in our next stage, what we are doing, that we are creating the beneficial microbes and giving them to the non-responders and trying to see whether they will respond. Next, please. Next. So this is, you can see how they respond. There's one patient, there was no, it was not operated at all. After giving the checkpoint inhibitor, the entire tumor went. So ultimately, because the theme was microbial technologies for this, so I actually chose this theme to show that how strong our bodily microbes are, and they could be used for the benefit of the this cancer. Actually, my mother also died because of this cancer in 6 months. They're deadly cancer. So 50% of them are surviving for nearly 5 years. Thank you very much for this opportunity.